good now. All right, so today we're going to talk about enzymes and energy. This is essentially two different worksheets from the worksheet packet that are sort of squished together. The enzyme one, or the energy one, we've already talked about a fair amount because it's covering the material on ATP, and there will be a little bit of stuff about that in here as well. So we're going to start off talking about just the basic forms of energy. There are 5 different forms. This is, physical or physics sort of definitions of the forms of energy, where we can have mechanical, chemical, light, electrical, and thermal. You could think of mechanical energy as the energy of movement. So the sodium-potassium pump uses mechanical energy, because it's actually shifting position in order to pump molecules from one side to another. Chemical energy is the energy stored in bonds. That's where we see it at least the most. So ATP has that energy in the last 2 phosphates, so in that bond. In biology, where we see light energy the most is the energy from the sun that comes in and helps to make glucose in photosynthesis. There aren't many organisms that can use light energy, but there are a few other ways that you might see it in biology. The eyes use light energy to a certain extent. But the energy from the sun and photosynthesis, that's most straightforward. Electrical energy we see in the form of ion movement, right, where we can donate that ion movement or the ion charge sometimes to other molecules. Those electrons, that's where electrical comes from. And then thermal is the energy from heat. And we'll show you a few specific examples of that in just a second. Okay. All right. So our first law of thermodynamics, there's only 2 of them that we need to know. Our first law of thermodynamics is that energy is conserved. Energy is conserved. And we can relate that to these forms of energy because we can convert one energy source to another, and this allows us to store energy. So another way that sometimes you hear this first law of thermodynamics is that energy is neither created nor destroyed, right? So we can convert mechanical energy into let's do it the other way. We can convert chemical energy into mechanical energy by breaking bonds in ATP and helping to move the sodium-potassium pump, right? So we convert one type of energy to another type of energy. Specifically, when we talk about storage of energy, we're really talking about the storage of chemical energy, and that's really what currency we're doing most of our energy conversions into or out of. We're storing them as ATP, or we're storing them as glucose. We're breaking down glucose in order to do other activities. The second law of thermodynamics just says that every time we have a chemical reaction, we lose some energy as heat. So when we do this reaction that we've talked about so many times, ATP is broken down into ADP and a phosphate, we can power muscle contractions. One of the things we can do. You'll learn the specifics of that in A&P. But as you break this, you're physically losing some energy as heat. It's coming out every time there's a chemical bond broken. We lose some as heat. In the case of muscle contractions, this is part of what makes you heat up when you're running, for instance. But it's also how we warm the body. If you get cold, you start to shiver. That releases some energy as heat, and you warm up a little bit. So those are our first and second law of thermodynamics. Those are the only ones that you need to know, right? We're conserving energy, and then we lose some as heat. Only two things, okay? But we do need to think about how we might use energy in the cell, because that's the focus of 160, is how do cells use energy. So a few of the specific things, I already mentioned this one, sodium-potassium pump, right? We use that chemical energy of ATP, and we break it down. So chemical energy stored in ATP is converted to mechanical energy to move ions in that sodium-potassium pump. So that's all that's happening here. You have a sodium-potassium pump right here. It opens up on one side. We attach some potassium, ATP, energy right here. We break it down, and then we can move this molecule physically and release on the other side. Right? So it's a protein that's actually changing shape. You don't need to know any of this stuff on the bottom. Ignore all that. I mean, technically you already know it, but big words. Ignore this. Okay. So some of the other things that we'll learn about some of the details of as we keep going forward. Nervous signals, right? So these are neurons that are firing. All sorts of muscle contraction and movement. Making babies. And also staying warm. More babies because they're adorable. And so why wouldn't you want to see more babies? Okay. So a lot of what we're going to talk about big picture-wise in our class is how we are going to reproduce. So how do cells divide? A lot of energy is required for that process. How are we sending signals? Energy required for that process. And then as you move on to AMP, what we just talked about with action potentials, that'll move into muscle contraction. That's another place where we use a lot of energy. Okay, and this is the basic way that we make and convert that energy from place to place. So we're going to start talking about metabolism. This is the basics of metabolism, but it's just a good diagram to see what's happening. You have this cyclic process where hydrolysis of ATP, so we're breaking apart ATP, releases some energy, and that releasing of energy powers whatever these things are, like riding a bike, right? Or it could help us to build molecules like proteins to make muscles. Okay? We then use those same molecules, that ADP that we made when we broke apart our ATP. We make ATP, and we have to put in energy to do that, right? So we have to eat things like an energy bar or glucose or starches from our food. So those things we eat, we put them in the ATP, then we break apart the ATP, then we make some ATP, and we just keep doing that over and over again. That's sort of the basics of metabolism. There's a definition. We'll talk about all the pieces of this. But think about molecules going from big to small, where the energy goes in, where the energy goes out, molecules going from small to big. So those are the sort of main components of metabolism. Okay. So our types of cell work, we're requiring this constant conversion to different usable forms, mostly ATP, and that is metabolism. So we're converting energy by chemical processes, and this includes both breaking down and building molecules. Okay? And we've already talked about breaking down and building through what reactions we use, which is that we can either do hydrolysis or we can do dehydration synthesis. And most of our metabolic reactions are some form of that, where we're either breaking something down, hydrolysis, or we're making something and doing our dehydration synthesis. All right. So a little bit of break on the metabolism front to talk about enzymes. So enzymes, we talked about yesterday, enzymes are proteins. They have a really specific shape, and that shape can bond to another molecule. Okay, so that's their structure. We pair that to their function, which is to catalyze reactions. Catalyze is essentially a sort of fancy word for helping it to start. And we'll talk about the details of catalyzing reactions in enzymes in just a sec. Okay. When this comes to metabolism, this means that organisms can then break down and build up molecules more efficiently and more quickly in most cases. Without enzymes, we would not be able to break down enough molecules to survive. So that efficiency, really, really important. We need to be able to break down molecules efficiently. Okay, so if metabolism is energy conversion by reactions, then this means that we need an individual enzyme to catalyze each of these reactions. Right? Enzymes can only catalyze one reaction, but they can catalyze it over and over again. just to say that one more time, enzymes can only catalyze one reaction, but they can be reused for that reaction many times. So over and over again, they can catalyze the reaction ATP into ADP plus P. Okay, so we categorize the reactions, or the metabolic reactions, that enzymes will catalyze in a few different ways. The first one just has to do with size. If a molecule is large and it's broken into small pieces, that's a catabolic reaction. If a molecule is small and we join it together to build into a large reaction, that's anabolic. So monomers into polymers, anabolic. Polymers into monomers, catabolic. It's easier to remember it if you remember that cats like to break stuff. Catabolic. My cat is very often shoving stuff off of tables and things, right? So they break them from big pieces into smaller pieces. We also have energy terminology that we have to deal with. Same reactions, but we want to know where the energy is going in those reactions, because we already know that reactions either will require energy or release energy. So we have two different terms that we use in relationship to those energy for each of those statuses. The first one is that we can have exergonic reactions that release energy. Exer like exit, just like exocytosis. And then endergonic reactions absorb energy or need energy. So we put that energy into the molecule. And that's what this figure is really trying to show you. If you have a catabolic reaction, you're breaking up molecules, energy is going to be released. That energy can then be used to make larger molecules. So one example of this might be that we eat some sort of food that has a lot of protein in it. We break that up into amino acids. They move into our cells. Those cells then use those amino acids to build some more cytoskeleton in our muscles so our muscles can grow. Okay? So we're breaking down, releasing energy, making other bigger things. Okay? And then we also look at how likely it is that a reaction is going to occur. So that has to do with the enzymes as well, but it's really focusing on this part on the energy, where we say that if it's a molecule that releases energy, it's thermodynamically favorable, meaning it's more likely to happen, whereas if it's endergonic, it's unfavorable. It's less likely to happen due to the rules of thermodynamics. It's harder to find energy. Any questions so far? This all makes relative sense. Okay. I know there's a lot of terminology here. So this slide just sort of puts it all together, right? Where we have exergonic reactions, they release energy, they are favorable, and they're also catabolic. Okay. But if we add the one more term that we know, those are also our hydraulic reactions, the reactions we do for hydrolysis. In endergonic reactions, we absorb energy, they're unfavorable because of that, they're anabolic, and they are dehydration synthesis reactions generally. Right? So all of those terms are just pointing at different parts of the same reaction. You should absolutely know how to relate those to each other. If I ask you or give you an example of a catabolic reaction, you should be able to tell me that that's, in all likelihood, a hydrolysis reaction. You should be able to tell me that it releases energy. Right? So make sure that you can put all of those terms together regardless of which one I'm talking about. So now that we understand the basics of metabolism, we're just going to talk about some specifics about enzymes. So these are just a few examples. And I'm going to point out something really important when we're talking about enzymes, and that is that when we name them to tell you what they do. Okay? So enzymes almost always, not always, but almost always end in ASE. Occasionally you'll see an SIN at the end. It's not quite as common, but ASE, almost always the ending. And then the beginning part tells us what it is that they're breaking down or putting together. So lactase will break down lactose. It's a type of sugar. Protease will break down proteins. Sometimes these are called pepsins. They break peptide bonds. DNA polymerase will make polymers of DNA. Right? So the fancy way of saying that is they add nucleic acid bases to DNA. So we're making a polymer. You can't necessarily tell by the name whether they're breaking down or putting together, though sometimes you can. But you should definitely be able to recognize that an ASE means it's an enzyme, even if you haven't seen that word before. We're going to look at a diagram that looks just like this in a second, so you don't have to write that down yet. Just showing you the basics of how enzymes work. Okay, so remember that we have reactants that go to products in any sort of reaction. Essentially what we have to picture is that enzymes work like a mechanical force that either shoves molecules together or it pulls them further apart, right? So if you were making a table and you needed to glue two pieces of wood together before you put a nail into it, then you might use like a clamp. So an enzyme might work as a clamp to shove molecules closer together. It means they're more likely to have this reaction occur, right? Alternatively, you could just pull it apart so it's easier to get to the internal structures that we want to break apart. So an enzyme could do that too. So it's all about how the shape can change. We say that that means they're putting stress on bonds, or it puts stress on atoms within those molecules. So this is just a basic graph of how this happens in terms of the energy of the reactions. Okay? So in this graph, we have a reactant, which is AB. So this is a polymer, or in this case, a dimer, that we want to break down into two monomers. So we're doing some sort of hydrolysis reaction here. Right? Any reaction, even if it's this kind of reaction, it's catabolic, it's releasing energy, it still needs some energy to get going. And that energy is called the activation energy. It's like the energy it takes you to actually get out of bed in the morning. You're awake, but can you really get out of bed? Well, that's activation energy, right? It's the energy it takes to actually get to the gym, not the energy to work out, but the energy to actually get there. That's your activation energy. Well, with any reaction, or without an enzyme, the activation energy is very high compared to the reaction with an enzyme, right? So that ability for the enzyme to change the shape of the molecule so that we're at this peak instead of this has reduced my activation energy by half. So that just means that this is far more likely to happen at a rate that will help us to sustain life. Okay? So otherwise these reactions would just be too slow. So activation energy, it's the energy to jumpstart a reaction. It's the energy we need to get started, not the actual energy to do the thing, but just the energy to get started. Enzymes can lower that energy for those reactions. Okay. So let's talk about the pieces of enzymes. They all have really specific names. We like terminology in biology, you know. So those reactants, when we're attaching them to an enzyme, we call them the substrate. The substrate. It's our reactant molecules. They fit specifically into an enzyme. It's the substrate, whether it's in one piece or multiple pieces, it's just the substrate. The place where they bind is called the active site. So you've got an active site down here, right? You've got your active site on this enzyme down here. So the active site is where the substrate will bind. Because of the shape of the enzyme, a substrate can only bond with a specific enzyme, a specific protein. That's true of any protein, really, that's bonding to other molecules. If it's a receptor protein, it can only bond to one specific place. If it's an enzyme, it can only bond to one specific place. So they have really specific jobs based on their structure. In enzymes, we call this the lock and key fit. You know, my key won't fit into your front door, but my key can be used for my front door over and over and over again. Right? So the lock and key fit, we can reuse enzymes, but only for the same lock, only for the same reaction. So in this diagram, that's essentially what's happening. You have these circular structures. They can't work with this enzyme, no matter how hard we try and fit them in. They just don't fit. Right? But they can be reused on this one over and over again to make a bigger molecule with those two circles bonded together. So this is just viewing it from a more three-dimensional structure. We have from here, we have this enzyme. It's an enzyme called sucrase. Right? So sucrase, we can tell by its name, has an ASE, so it's an enzyme that's probably going to attach to sucrose, where OSE is almost always a carbohydrate. So we have an enzyme, we have a substrate. They're going to come together, and they're going to make this enzyme-substrate complex. A complex is just a complex molecule, so two molecules bonded together. Once the substrate has bound to that enzyme, then we can add H2O. So we're doing a hydrolysis reaction. We're adding water to break apart. We've now broken those two molecules apart with a smaller amount of activation energy. At this point, we call this a product enzyme or enzyme product complex. Because now we don't have a substrate anymore, we have a product. And then we release those from the enzyme. We now have fructose, which is a monomer, and glucose, which is a monomer. They're the broken down pieces of my sucrose. And my enzyme is available again, so then I can reuse it to do the same reaction over and over again. Okay. The reason why we care about this, enzyme shape, is very, very important to its function. Okay? We've talked about protein structure over and over and over again. Enzyme shape is very, very important to its structure. Enzyme function really is dependent specifically on the active site. We also know that enzymes work better when they're in a warm and very specific environment. But that environment is really important. It has to be stable. So environment matters. Now, hopefully next week we'll have some time to get into the lab and do a little bit of an enzyme experiment. Really depends on how many more snow days we get, but we'll see at least different temperatures and how that might affect enzymes. The basic idea is that enzymes work better if they're warm. They don't work as well as if they're cold, which is why refrigerating things is a good idea. That slows down the reactions of bacteria that are catalyzed by enzymes. And so that means our food is less likely to get covered in bacteria that might make us sick. Okay. There are some ways that outside of the environment we can stop enzymes from working. Some medications work this way, but we also can inhibit certain enzymes and then start them again. So this process is called inhibition. Because enzyme function depends on its active site, the easiest way to inhibit an enzyme is to cover up its active site. However, we could also change the shape of the active site, and that's actually a little bit more common. When we cover the active site, so let's say we have an enzyme like this, and we cover it up, we call this an antagonist. It's just something that covers up. When we're changing the shape, though, that's what this picture is over here. This is a type of inhibition called allosteric inhibition. You do have a problem on this in your worksheet. You have this enzyme where here's your active site. Okay. There's an allosteric site that's just across. It's another part of the protein. And if an inhibitor attaches to it, then now the active site won't be the correct shape for the substrate. The substrate would have fit up here, but it doesn't fit over here. So all we've done with allosteric inhibition is change the active site. Okay. So we could use allosteric inhibition or we could use an antagonist. Either way, we're making that enzyme non-functional. Most of the time, though, those aren't permanent. We can take off that allosteric inhibitor or the antagonist and we can have it work again. Sorry. A more permanent change is what we call denaturing. Denaturing of enzymes unravels them and stops them from being able to function normally. There are lots of different ways to denature enzymes. The ones that we encounter the most have to do with high temperatures and then using pH or other ions like salts. Okay, so let me just show you essentially how denaturing works, depending on what kind of item is denaturing. So we've got a fully functional protein over here. So that's in its quaternary level, right? So it's got all sorts of different coils, beta sheets, the alpha helixes, etc. In between each of these places in structure are hydrogen bonds, or sometimes ionic bonds, those hydrophobic, hydrophilic interactions, etc., right? So if we've got all of these different little places where there's hydrogen bonds, if I take something like, for instance, a sodium that has a full charge, just like when we talked about water, and I said if you add sodium to water, then it can break apart the water molecules so they're moving around a little bit more, it melts the ice. Well, the same thing can happen here if we stick this plus in there with all those partial charges, and we do that to a bunch of places, then we can start to unravel this protein. Okay? When it unravels, it forms this sort of loose structure where you cannot reform all of those many, many, many hydrogen bonds that were all over this structure before. It's like if you've ever knit and you make a really perfect ball of yarn, and then your cat gets to it. I know I like cat stories, right? And your cat gets to it and just pulls it apart. You might be able to put it back into a ball, but you're not going to get it into this ball, and this ball shape is very important. So as soon as those proteins have moved out of their quaternary structure, it's permanent, it's unraveled, it's broken forever. Okay? Now, temperature can do this in a slightly different way than salts can, because essentially when you get too hot, things start to move more, they vibrate faster, and that just starts to break apart bonds. So temperature, when it's high, is good, but when it's too high, it denatures. And, if it's too low, then it doesn't denature, but it's slow. Sorry, that's a little messy. So the reactions will go slow, but it doesn't denature them. Okay? Think about this in terms of food preparation, right? I told you we keep things in the refrigerator because it slows bacterial enzyme movement, right? So they can't reproduce, so we don't end up with E. coli all over our food. But if you leave it out in warm temperatures, it'll start to reproduce. So what do? We cook it. And if we cook it to a high enough temperature, we can kill off the enzymes, denature them. Those bacteria will die, and we can safely eat that food, right? If you just put it in the freezer, it will, at that moment, not allow those bacteria to grow. But then as soon as they warm up again, they can grow again, right? It is possible to freeze things enough that they die, right? Humans will die eventually. We do need movement. We do need whatever. But it's not a set limit. There are also differences in specific enzymes in specific organisms. So for instance, the bacteria that are in your stomach have tolerance for H plus ions, for acid, that enzymes from bacteria that live on the surface of your skin don't need, right? So we do have adaptations, those buffers we talked about with the acid-base stuff. Buffers can help us to deal with acidity or too much basic. Enzymes can, in bacteria, be covered by those same processes. Does that make sense? Okay. So when you're thinking about denaturing, it's salts or any other ions, pH, H plus ions, right? Even base, OH ions, that would work too. And then temperatures if they are too high. Think boiling, because boiling's good for us because it means we can, eat things or drink things that otherwise have bacteria in them. Any questions about denaturing, enzymes, how enzymes work? Okay. So I'm going to end this recording right now because that's all I can do on this. But we're going to pull out your worksheet packet. I'm going to let you take just a 5-minute break, and then we'll regroup. I'm also going to check the weather. Okay. 5 minutes. 5 minutes.